Thanks for watching episode number 127 of the Doctor Podcast Show, and I'm your host, Dr. Robert Cykiert. Please subscribe and follow Doctor Podcasts and like and repost this episode. We'd really appreciate that, and it'll help me create more excellent programs like we have today. Today's show is on a very important and fascinating topic that I think will have a huge impact on the practice of medicine in the future. It's about same-day whole genome sequencing of human DNA, which is incredible. We have three great guests and experts on this topic today to explain it all to us. First guest is Mark Kokoris. He's the head of SBX technology at Roche Diagnostics. And by the way, Roche Diagnostics is a division of the parent company Roche, which was founded over 125 years ago and has grown into the world's fourth largest pharmaceutical company by revenue. It's also one of the largest biotech and diagnostics companies and a global supplier of transformative, innovative solutions across major disease areas. Roche focuses on preventing and curing diseases with the highest societal burden, with the goal of improving health outcomes and reducing costs for patients and healthcare systems. Now, Mark Kokoris has nearly 30 years of experience working on the cutting edge of biotechnology, conceiving and developing innovative and transformative technologies. His vision of creating a next generation DNA sequencing technology that would enable broader use in healthcare led him to co-invent the SBX technology in 2007. That same year, he co-founded Stratos Genomics, where he became CEO, and in 2020, he led his company through the acquisition by Roche, where the SBX chemistry could be further developed. Mark, who heads all SBX research at Roche, is an inventor and has over 20 US patents. Our other guest is Dr. Niall Lennon. Dr. Lennon is the chair of the board and chief scientific officer of Broad Clinical Labs, the Broad Institute's wholly owned subsidiary that develops, implements, and validates innovative technologies that enable transformative advances in scientific discovery, clinical research, and diagnostic medicine. He has been with the Broad Institute for over 20 years and has contributed to the development of applications for every major next-generation sequencing platform across a range of fields. Dr. Lennon built Broad Clinical Labs to facilitate return of results to patients and to support clinical trials. Incredibly important. Our other guest is Dr. Monica Wojcik. Dr. Wojcik is a neonatologist and clinical geneticist at Boston Children's Hospitals, which is one of the top children's hospitals in the USA. She's an assistant professor in pediatrics at Harvard Medical School. She specializes in rare diseases affecting the fetus and neonate. She has post-pediatric residency fellowship specialty training in both neonatology and genetics, and subsequently obtained her Master of Public Health degree to augment her clinical effectiveness and health services research. She currently serves as the director of the neonatal genomics program at Boston Children's Hospital and is the medical director of the Manton Center for Orphan Disease Research. Dr. Wojcik research focuses on the application of genomic medicine in the perinatal setting, particularly relating to genetic diagnosis. So I want to thank all three of you for taking time from your busy days and schedule to join us today to discuss one-day whole genome sequencing, which is really incredible. Monica, I want to start with you. You were the lead author in an important recently published article in the prestigious New England Journal of Medicine about same-day genomic sequencing in newborn infants. Now, you see these tiny patients in the NICU, which is the neonatal intensive care unit, every day. Can you tell us about one baby whose life was changed because doctors finally figured out the genetic cause of the baby's medical problems? Even if it took weeks to do that genetics testing and evaluation in the past.
That's such a great, a wonderful and timely question, actually, because the degree of impact depends actually on the timing of result. You know, you imagine if the result's taking weeks, the life's already changed by the time the results are back,
Right.
So there are certain highly invasive procedures, like organ transplant, that can be life-saving, but we wouldn't want to proceed with that unless it was clearly indicated because of the potential, obvious risks. So one example I can think of is, rare genetic pulmonary respiratory disorders where babies are born with such severe lung disease that they're dependent on a ventilator and sometimes need to live on ECMO, on bypass. And it's really hard for the body to live that way. And the only way to get off of that would be a lung transplant. So anything
Wow.
you can do to prove they have, a genetic lung disease and not some other non-genetic condition that could spontaneously improve over time, that can shorten the time to transplant and improve their outcomes. So in the past, we've had babies that didn't get a transplant that they needed until they were several months, even close to a year old, and sadly others that haven't survived to that point. But recently, we've been able to make that diagnosis in 2 to 3 days and shorten that window before transplant to weeks to months. And that's such a tremendous benefit that we feel very lucky to be able to provide now.
So by getting the genetic testing, you're sure that this is a genetic disease and not some other issue that'll get better on its own.
Exactly, that's right.
Wow, that's amazing. So, Mark, congratulations on being co-author in the New England Journal of Medicine article. Many people think that DNA is just a twisty ladder, what we remember from kind of high school biology. In recent years, though, people now understand that DNA is billions of molecules arranged in a specific sequence and configuration, and it codes for our genes and our chromosomes. Can you tell us what a genome is and what genome sequencing is?
Yeah, that's great. Thanks, Robert, and thanks for having us today. This is great. Fun
My pleasure.
conversation. So, yeah, a genome is essentially the complete set of DNA from an organism. So it contains the instructions for development and function. It's essentially like a master operating manual, if you want to think of it that way. So when you read someone's genome, you're determining the precise order or sequence of the DNA code, which is composed of four molecular building blocks called nucleotides, which are often referred to as in shorthand as A, C, G, and T. That's usually how everyone refers to the chemistry there, A, C, G, and T. So to sequence a person's genome, you'd isolate a sample of DNA, either blood, tissue, swab, other means of isolating DNA, and then use a technology like the one we'll talk to here, SBX, to actually sequence their genome. And this information, is Monica and Niall know, is useful for understanding predisposition to disease or helping manage complex diseases.
Right. Now you mentioned ACGT as the molecules. How many billions of those molecules are there that make up our entire DNA?
Well, the human genome is about 3.3 billion bases or nucleotides together,
Wow.
strung together as one big information storage system, as I like to call it. Yeah.
Right, almost like computer code. Now, what does it actually mean to read someone's genome, Mark? What does that mean? What do you do when you're reading someone's genome?
So actually, that's the process of sequencing and measuring the sequential order of these molecules. So, the first draft of the human genome was actually published in 2003. It took 13 years and the cost of $3 billion to generate
Wow.
that first reference genome. So we've come a long ways in our ability to sequence a person's genome. And
Right.
so, and now the pace of this technology innovation has been tremendous. I think it's outpaced Moore's law in terms of the integrated circuit doubling time. Sequencing, I think, has actually outpaced that. So, and this led to dramatically improving efficiencies, driving down costs, and reducing time necessary to sequence. So, the Roche technology we use here, it's called sequencing by expansion, or SBX, and we can talk about that later if we'd like.
Yeah.
Brings new capabilities that allow for even faster, more efficient sequencing. So the work we did here, we sequenced the human genome in less than 4 hours, which, to use the scientific term, is bonkers, right? I think that's what we can agree is probably a good,
Yes.
yeah, a good term. So, and how much, yeah, faster could we do this? I mean, that's open question. The actual sequencing time only took about 15 minutes. It was the other steps before and after.
Wow.
that add up to the 4 hours. But I also want to say one thing while Niall and Monica are here is what great teams this was for this project and what fun this was. So my team in Seattle did an amazing job of compressing all these workflow steps necessary to do the fast sequencing. Our computer science guys compressed all the algorithms for this, and of course Broad and Boston Children's, like really top notch, a wonderful group of people to do an exciting project. And I think on one personal note, I have to say, I had a nephew, Ryan, who was born with chromosomal deletion, and it was 20 years, over 20 years ago. And just seeing how the uncertainty, all the biopsies and endoscopies, all the things they had to do because they just didn't know what was going on with Ryan, and having the idea that into the future we're going to be able to do these kind of things more and more often is just awesome to be a part of. And so I want to thank Monica for what she does and Niall for what they do as well.
Right. So let me summarize that. What used to take 13 years now takes it's 4 hours. But that's really incredible.
Yeah.
Now, Niall, congratulations also on being the co-author of the New England Journal of Medicine article. Now, you run one of the world's biggest DNA sequencing labs at the Broad Institute. How long did it take to sequence a few full human genome when you first started doing this? And what did it cost back then to do that?
Yeah, thanks, Robert. As Mark mentioned, the very first human genome, was actually sort of the technology didn't even exist to do it in a single lab when this project started, and it needed to be an international consortium to do it, and that completed about 2003. I joined Abroad in 2005, just a few years after that first draft was published. And that was really the beginning of what we would sort of say is the genomics revolution. The initial human genome project really, like a space race, sort of kicked off this wave of technological and computational development that led to newer technologies being developed and that allowed us to actually start to do human genomes in single labs and ultimately, in single days. So when I first started, it really still wasn't possible to do a sort of a human genome in any kind of, cost reasonable way. A few years on, in sort of 2006, 2007 timeframe, we started to see new technologies be developed that allowed us to start to think about doing human genomes more frequently. But it was still even a few years after that, into the 2010s, when the cost came down to be tens of thousands of dollars at
Right,
that time, that we started to do more human genomes more routinely, and it would still take us many weeks to do them.
right.
Um, and fast forward into the last few years, now we do sequence human genomes somewhat routinely. In fact, in my lab this year, we'll probably do about 170,000 human genomes
Wow.
to serve the research community and the clinical translational community that we work for. And those genomes today cost a couple of $100. And typically, if we're not rushing them, again, they'll take several weeks. More, not that the sequencing time itself takes several weeks, but that the process of getting the samples in and pushing them through takes several weeks. So we went from many, many years to now in routinely in sort of the research setting being several weeks and being a couple of $100. You would say it's probably typically under $500 today to sequence a human genome just from the data generation part of the process.
Wow, that's incredible. Now, Monica, you touched on this briefly before. In the past, parents might wait 2 to 6 weeks or longer for genetic results so that you can figure out what to do for their baby. What does that wait feel like in the NICU for doctors, for you, and for the anxious moms and dads of the baby?
Yeah, it's such a great question. And, families come to our NICU under all different circumstances, and some know, from prenatal testing that their baby will have medical problems. But, if we think about the families that were entirely unsuspecting, so they thought they had a healthy pregnancy, a healthy baby, and now they're here in the NICU and their baby has persistent seizures, or they can't breathe, they're on a ventilator, or they need emergency surgery. I think for many families, this is the scariest, absolute worst moment in their lives as parents, and they're hearing from doctors this wide range of possibilities. There could be healthy life, there could be potentially death, and parents and doctors are trying to make these important decisions really in this void. So, you can imagine there are families that are just hanging on, waiting for this result while their baby is on life support. And that's, an uncomfortable feeling to say the least for doctors and patients alike. And for me, those are the ones where I'm calling the lab, emailing every day to say, can you tell me anything yet? We're
Right.
just waiting for this to go to make the next move. So, it's a challenging situation to be in for parents. I can't even imagine what they go through.
Right. So this has made things much better for you and for families as well. That's really great. Now, in your study that I read, there was one baby who got a same-day answer that explained their seizure disorders. If that result had come back in 6 hours or 4 to 6 hours instead of days, how might the treatment have changed for that baby?
Yeah, I mean, I think even though our current rapid genetic diagnostic approaches are very good, as a neonatologist, I also want to acknowledge that our clinical diagnostic skills are pretty decent as well. So when the results are coming back from the genetic test in days to weeks, often we already sort of know what we're dealing with and have initiated
Mhm.
appropriate treatment even by the time the results are back, which is what happened in this case. And remember, this is a research study, so we were trying to time these results for a period when we'd have the clinical result returning around the same time as the same day result. So this is all somewhat hypothetical, but given that there are certain genetic epilepsy syndromes that have
Hmm.
particular treatments that are the most effective, if we had the results the day that the baby started seizing, we could use the most effective drug, as that first line therapy instead of starting down our usual pathway that involves trialing different agents to find one that works. So it could really shorten that, therapeutic, the time to effective therapy as well.
Right. So this is customized, personalized medicine at its peak, basically.
Yeah, absolutely. Yeah.
That's incredible. Now, just theoretically, if you can do a whole genome analysis in a few hours, could you in the future possibly theoretically use something like CRISPR or other DNA editing to actually fix the genes?
I think in terms of the gene therapies, CRISPR, et cetera, we have a very exciting future ahead of us, and not everything can be completely reversed. But I do think
Right.
that, one important aspect of these gene targeted therapies is that you really need the precise genetic diagnosis to access them. So I
Right.
think our job and the power of this collaboration, especially was to make these diagnoses as fast as possible so that our patients are eligible, and that's, I think, the incredible potential that we have here.
Right. So, Mark, now Roche just published this breakthrough in the article that we've been talking about. Let's dive a little deeper into this amazing Roche technology. Roche and you invented something called sequencing by expansion, which you mentioned earlier, or SBX. Can you explain what that is for the average person in the audience who has great interest in the latest medical technology and advances, but not that much expertise or knowledge? Basically, how does SBX work? Tell us a little bit about the XNTP molecules and the nanopores, and how do you go about translating a sample like blood into DNA information in 4 hours?
Yeah, no, that's great. Now, this one I can talk about for hours, and I think I actually have sometimes. I kind of can go on and on. So I'll try not to go too much into the chemistry here and try and cover it and maybe use a couple examples. So, I mean, basically, our approach to efficiently sequencing DNA is to not sequence DNA. And that's kind of a weird statement, but and it'll be clear later on. So the four nucleotide bases I referred to before, A, C, G, and T, make up DNA. They're spatially compact information storage system, as I called them before, and the bases are actually structurally quite similar. So this basically means this is actually a really hard measurement challenge. If you have all these bases, you have to do this resolution. How do you do that efficiently? These are sub-nanometer spatial resolution. So this, from a measurement standpoint, is extremely difficult. And every sequencing technology has to resolve this, figure out how to solve this problem. So if you think about small font in words that are spaced really close together and you can't see them. Now imagine a bigger font with better spacing, like
Hmm.
on my wife's cell phone, for example. She's going to kill me, by the way. Biggest font I've ever seen in my life. But it's a great example because you can actually resolve the large font. And so, yeah, and it's similar with SPX. So we essentially increased the spacing or expanded the structure so we can measure and detect these more easily. So to do this, we created a biochemical conversion process. And so this used enzymes and chemistries that never existed before. And you'd mentioned the XNTP, this expandable nucleotide. So it's not a regular nucleotide, it's an expandable one. And we use that to create this surrogate molecule called an expandamer that's a surrogate of the original DNA. So now it's no longer DNA. It's this new molecule that's highly engineered that's about 50 times expanded relative to regular DNA. And the idea is that, and I got a picture behind me. The idea is that now you can measure this molecule a lot more efficiently at higher speeds. And so now you see it all kind of coming together here. And so the way we did that was we brought another technology together called a nanopore sensor array. So the idea is you use these small protein pores where you actually thread the DNA through the pores and you can measure electrical signal changes of this long expandamer molecule and do that really, really efficiently. So this is like radical technology to be able to do this at the speed and throughputs that we do here. And all of that works hand in hand with accelerated compute and analytical methods of how you actually take that data streaming off very efficiently. So all of it kind of comes together. So with SBX, we essentially rescaled the structure of the molecule to solve the problem. So it brought on tremendous chemistry challenges, biochemistry. These things were never done before, molecular engineering. And honestly, we had to break a lot of rules to make this work, but I'm really glad we did because once we finally figure out how to do it, and it was 2007 we started the company, it actually has a pretty amazing outcome.
Wow, that's incredible. I love that analogy of increasing the font so you have more spaces. It really explains it well. Now, my understanding is that you began working this in your startup company before Roche bought your idea. What was the aha moment that made you think same-day sequencing was possible? When did you realize that you were on the brink of this incredible discovery that's going to change the world eventually?
Yeah, that's a great question. A little bit about the startup company. So I co-founded Strauss in 2007 with my friends Al Steffen and Bob McGruer, and they actually had an engineering consultancy. Al was the CEO of this engineering consultancy. It is called Stratos Product Development. So you see where the name came from. Bob was the CTO. And so I brought the idea to them. We knew each other through work and collaboration. And so at some point, we brought this kind of wacky idea about changing the DNA molecule, and it was just this is one of the things that history, the three of us coming together, I think we all wanted to do something that was game changing, and we saw it in this because we saw about how it solved the measurement challenge there. And so the way we looked at it, honestly, the speed, the flexibility, these are all rooted founding principles of SBX. So it really wasn't an aha moment for us, really. It was more about, the evolution of the technology to get to this point. But I'd say probably about 2 years after the acquisition where SBX came into Roche, when I was seeing the throughput, seeing the capabilities coming together, I thought, okay, we're probably going to be able to do this. And that's pretty cool.
Wow, that's amazing. Now, Niall, there are commercial labs that offer fast DNA tests in days, but not hours. Why wasn't that fast enough? And what convinced you to team up with Roche on this amazing new 4-hour version?
Yeah. So as I mentioned earlier, most of the genomes that we do, the tens or hundreds of thousands of genomes that we sequence per year, are really in service of discovery or research, research into certain diseases, disease foundations, federal awards, and pharmaceutical companies. And for the most part, those studies really are looking at data in aggregate. They're looking at large cohorts of patients or disease individuals. And so they don't really need their genome to be done in hours. So that's how sort of most of our genomes are done. But over the last several years, there has been a growing appreciation that rapid genome sequencing could be useful in certain contexts, one of which is the NICU. And this was advocated for by many groups, but most notably, I would say, in our community, Dr. Stephen Kingsmore at Rady Children's Hospital in San Diego has really been a trailblazer in this area. It was Stephen, in fact, who set the first world record for doing whole genome sequencing in the NICU setting. Subsequently, Dr. Ewan Ashley at Stanford had, using a different technology, set a newer record for how quickly you could do the DNA to variants from a human genome. And that was the record that we actually beat in this project.
Wow.
So, however, despite those sort of demonstrations, which were really, seminal in sort of showing what could be possible, the difference was that the way they were done wasn't really reproducible in the everyday setting. It was done in sort of a heroic way to show what was possible, but the technologies and the process and the cost associated with doing it that quickly weren't really feasible to do in an everyday setting. And so, as we were working with Roche on this new SPX technology that Mark invented, as we started testing it in the lab and realizing that it could be done at this speed, not as sort of a one-off heroic effort, but actually that was just the way you could run it. And with certain tweaks that Mark's team did, we could do it even faster. In our group, we were coming into that with the context of knowing the work that had come before, and we said, gosh, actually, we think we could show that this could be done routinely at this time, at this kind of speed. And if you could do it routinely, that might actually make a difference to people like Monica who need to see this data routinely. And so
Right.
that was really where the collaboration came together, where we had been working with Roche on the technology evaluation and then reached out to our colleagues at Boston Children's and said, look, what if we were to demonstrate that we could do this not just once, but actually over and over and over again over a period of time to show what's possible once this technology is more widely available, to bring down the time and get you the results on the same day. And as Monica has described, in their setting today, they've modified or at least they think about their clinical workflow with the assumption that the genetics data will come back in this sort of 5 to 7 day period. And so you kind of work around that assumption.
Right.
But actually demonstrating that we could do it on the same day completely breaks that assumption. And going forward, if we actually make this routinely available, then I assume, and Monica can sort of say, I assume that the actual thinking about how to use this information will change as they think about these cases going forward. And so that was really the context of how, which is essentially our day jobs at Broad Clinical Labs, is how do we work with really exciting novel technologies and then bring them to the clinical genomics world so that they can make a difference.
Right. So it's a team effort. It's Roche and you and your labs and the doctors,
Right.
and ultimately the patients benefit. Now, Monica, this is currently still a research tool, but if it becomes a regular everyday clinical tool in the future, and possibly every NICU eventually has this available, what's the first thing you'd want to tell parents on day one about this?
Well, I think the message in some ways is really still the same, that, our job as doctors is to find out everything we can, do everything we can to figure out what is happening to their baby, to find the explanation, and, we're there for them every step of the way. I think we think about the immense importance of same day results and how they can influence these really critical care decisions. But, I think for the parent's journey, in some ways it really starts on that day that you receive the diagnosis, and then you have to figure out what to do beyond that. So that level of support and kind of just working through it is going to be so critical, and perhaps even more so if we're actually coming in with a diagnosis on the same day that, or maybe the day after we even bring up the idea of a genetic condition. So this is a lot of information for families to process, and I do think that message that we're there as clinicians to really help them work through it is not to be lost in this process.
That's great. Now, Mark, regarding Roche's role, you didn't just speed up DNA reading here. You also focus on quality and thinking about lab scenarios. Why is that important? Why does that matter?
Yeah, I mean, for the fast sequencing that we did here, we had to reduce, as I mentioned before, all the workflow steps in order to get to this result, and at the same time maintain quality. And I was really happy to see the quality of the sequence. We won't go into technical details here, but it was actually quite good, the quality that we got out of this sequence. So that was, I think, really an important part of this, that we hit that standard. But a couple thoughts on lab scenarios. So SBX will launch as a research use product, and we'll call it the Excelios platform, is what it's called. That'll happen next year. And I'm really excited. I think this is something I've been thinking a lot for many years about. I'm really excited to see what researchers do with these capabilities and how they bring this capability of SBX to bear. So, of course, faster results for time-sensitive applications. This is great. But then also thinking about ways to implement SBX at scale, whether it's a 24/7 operation or running more flexibly for low volume users in labs that don't have that kind of throughput. That's one of the strengths of being able to flexibly, lab scenarios where you're flexibly running and doing sequencing. And then really rethinking in this format with SBX how to improve operational efficiency. So those are a lot of the things I think about. Or, on the other side, testing new applications. Like there's things we haven't thought of. And when you have a new tool, you can think about, other ways you can sequence that, either you hadn't thought of before or you really needed a new technology to enable. But then one last thing on the lab scenarios, I believe there's a cadence and momentum to science and innovation. And I'm inherently impatient, as my team knows. I want to get results
Right.
faster and always. But I do believe there's a momentum to innovation and information coming back to you, that having that flexibility and ability to sequence faster and get information in a timely way can maintain at a research level momentum, because it's not inevitable that innovation and ideas will come to you. I mean, I can think about all of innovations we've made. You had to be ready for them when they came. And I think there is that timeliness that I'm really excited to see how people leverage once the technology gets out there.
Right. Now, Niall, you tested this on real NICU babies, not just lab samples. What was the most surprising thing that worked or that didn't work when you tried it in the real world?
Yeah, I think one of the most surprising things was it actually worked so well. We are fairly used in our group to trying new technologies, and as you said, oftentimes the things we try on them first are really control samples or reference samples, samples that, really high quality DNA where we know the answer, and those samples can work well. However, the big leap for many new technologies is actually being able to use them on real world samples. And real world samples aren't as neat and clean as these laboratory samples.
Right.
When you get blood from a baby and when you get different types of specimens, things can happen. The DNA might not be as high a quality, or there might be things in the blood that are not in your control sample that throw off the technology. And
Right.
actually, we didn't see any of that. And so, really, going from the control samples to the real world samples, the quality of the data, the utility of the data was no different. And that was in itself really, really surprising. And I think the other part with any new technology, with all respect to Mark as well, we are used to new technologies coming in, and then like it works great one day, and then the next day it breaks, and that's just we're okay with that. We get early access to these technologies to help companies kick the tires and find out how they might not work. But again, here in this study, we over the course of 3 weeks, we were doing samples, every day, every other day, and we didn't have those sort of technical issues.
Wow.
It worked when it needed to work, which for a technology sort of pre-commercial like this, that itself is sort of surprising and great to see and very encouraging.
Wow. So, Mark, when are you getting your Nobel Prize for this?
It's funny, actually, two of my old colleagues from my first company won the Nobel Prize last month. So
Oh, really? Oh, all right.
yeah, we had a nice party for them. So yeah.
That's awesome. So, Mark, now Roche spent years and I assume millions and millions of dollars turning your idea into a reality. What part of this technology is uniquely Roche and that's something that no one else could do but Roche?
I mean, well, first, the most important part was Roche brought two companies together, and they brought Stratos Genomics together. with SPX chemistry, and then Genia. Genia was a company that had the sensor array technology I referred to before.
Mm.
But it's really not surprising that we ended up at Roche. I mean, PCR, they brought PCR to the world, and PCR was kind of my inspiration as a young scientist. I looked at that and I was just blown away the first time I did PCR to amplify DNA, and I just thought, okay, this is amazing. So I'd always had that in mind, that Roche would be a great suitor for this technology, and they've certainly brought uniquely facilitated this coming together of these two, and I call them historic technologies because they really amazing chemistries, amazing what Genia did in terms of how they solved the sensor module challenges, but then coming together. Roche did a fantastic job of bringing those together. They have a long game look when they bring technologies together. They have a long view of that, and then certainly have the global capabilities to broadly bring SBX to the market. So I think all of those things were in mind.
Right. Now, Mark, when you walk into a research lab in 2026, next year, what does the Roche SPX machine look like? How big is it? And can you describe it for us?
Yeah. I mean, well, I hope it's well used, looks well used. That's what I'm, yeah. And I expect there'll be a lot of enthusiasm around using this new technology, learning, I think, new workflows. Again, there's some amount of learning how to sequence differently, because it is a very different approach. And this is a good thing. And as far as how it looks, it's essentially two tabletop systems. One is the sequencer, and one is the actual unit that does that SPX chemistry, that conversion step that I talked about before. And you can see, if you go to Roche sequencing, you can see a website that we have, and you can see all kinds of information on there that shows images of what the instruments look like, and has a lot of background of the technology.
That's great. I'll include that in the podcast. Now, Monica, doctors usually aren't that comfortable trusting black boxes when they're taking care of very sick patients. How did Roche prove to you that their 4-hour DNA result is just as accurate as what used to take 5 or 7 days in the past?
Yeah, well, I should say that, the opportunity came to me via the Broad Clinical Labs team, and I was already familiar with several of their team members and their work from my own work with the Heidi Ream at the Broad Institute. And so I've always thought very highly of Niall and his team. So when I think it was Sean Hofer and Katie Larkin from Broad Clinical Labs told me that they thought the genomic data coming off this new Roche machine was high quality, that was honestly pretty good, is good enough for me. But we did send them 5 cases that we knew from Boston Children's, from the Manton Center, that we knew had diagnoses to make sure they could find them, and then they did. So that also helped.
Wow.
And then finally, in our study, we ran clinical testing through our usual clinical lab in parallel. And those results also matched what the Broad team had found. So none of that was surprising to me, but because, I think it was a collaboration that I was very much looking forward to with the team that I value and trust. So I didn't really have many doubts, I can say.
Wow, that's amazing. So you must have been like amazed with the results when they were identical to the older technology, right?
Right, but so much faster.
Wow, yeah, huge difference. So Niall, Broad Institute is a nonprofit research institute, and Roche is a giant international, highly respected pharmaceutical company, diagnostic and medical device company. How does that partnership actually work, the two of you working together?
So at the Broad, we've been in the business and at the forefront of evaluating new technologies for sequencing DNA, for as long as I've been there, for the last 20 years, since these technologies first were invented. And as I mentioned earlier, we are one of the largest sequencing labs in the world. And so we are really good at understanding what makes a good technology and a bad technology, and also really thinking about there are many sequencing technologies, and they're sort of fit for purpose for lots of different applications, and we serve through our community, our academic community at the Broad Institute and at our affiliates at Harvard and MIT. We are used to generating data across a range of application areas, from infectious disease, common disease, rare disease, and cancers. And so because of that, because we have a really active user community who are making great discoveries using this data, we are often one of the first groups that companies who have a new technology for sequencing DNA come to
Hmm.
and ask us, would you be interested in working together and helping us and thinking about the applications of our technology? We don't always say yes, but certainly when Roche approached us, we were very excited to say yes. As we heard more about the technology, we got even more excited. And
Right.
as we got our hands on it and started to see what it could really do, we got even more excited. And that's when we brought in Monica and the team at Boston Children's. So we were very excited that this partnership worked out.
Yeah, great story. Now, Monica, currently, again, this is a research tool, but do you see possibly a day in the future where every newborn baby gets a full genomic sequencing at birth so that pediatricians are alerted to potential medical problems that the baby will face as a child and adult? Because it's good to know this information as early as possible. Do you think that's a potential use in the future?
Yeah, I think so. And there are some large studies looking at that very issue. I do think, one important distinction I want to make is between, genetic diagnosis and screening, because I think a lot of people hear, genome sequencing and they assume genomic screening, but that's really where you're looking at the genome of a baby or anyone who's healthy, asymptomatic, and you're trying to find diagnoses they weren't aware of that could cause future disease. But
Hmm.
I kind of want to contrast that to studies like ours where we sequence babies that were already symptomatic
Right.
to try to find out what's causing their symptoms. And I think the distinction is important because the way you go about looking for reporting variants is different in kind of the diagnosis, versus screening realm. And because our knowledge of genes and variants that cause diseases is incomplete, for screening purposes, you try to restrict what you're looking for. Whereas for diagnostic purposes, we tend to cast, a much broader net.
Right.
And then also just the psychosocial implications of receiving a diagnosis for your baby who's already sick compared to one that is healthy at the time, are
Right.
quite different. But I do think that regardless, more and more babies are going to be getting sequenced. And in fact, we have a protocol at Boston Children's in our NICU right now to offer rapid genomics universally for babies that come into our NICU for diagnostic purposes. So I really hope this availability will spread, especially for the babies that we care for, in our NICUs that are already sick.
Right. Do you know if this would potentially be covered by insurance in the future,
So
or
that's a
if
complicated question.
yeah,
It's definitely reasonable to say that earlier diagnosis is better. I think given that the cost of the faster techniques tends to be higher than the slower ones, again, all these are much better than they used to be. I think it might be a hard sell at this point for insurance companies to pay for, a same day test outside of an ICU setting, as Niall had alluded to earlier. But it's
right,
an interesting question to ask, who needs the diagnosis same day? Who can wait 2, 3 weeks? Even in
right,
an ICU, it might not be one size fits all, but I do think, payers are coming around certainly to covering rapid genomic sequencing for critically ill babies and children. And I hope this would similarly, be included.
right. Now, Mark, again, it's a research tool, but could same-day DNA sequencing be helpful for adults as well? For example, could it help you predict somebody who's predisposed to certain types of cancer, heart attacks, strokes, infectious diseases? So is there a role in the future possibly for doing this on adults as well, the same-day genetic sequencing?
Yeah, I mean, I think you can imagine a range of time-sensitive applications that could emerge using SBX. I think this was a great one, a great example of that. But I think certainly infectious disease is an interesting area. But I think there's quite a few that will emerge that people like Monica, Niall, and others will come up with. And so having that tool there, I think sometimes when you make a tool, people will be motivated to use it in a certain way when it has that capability. But I'd say our focus is the same as it has been at Roche, is to continue to develop capabilities, enhancing those capabilities, and just thinking about speed and throughput, efficiency, cost efficiencies. Read length is one we didn't talk about, reading longer fragments than the ones we currently do. All these type of things will continue into the future. And I like to say we're just getting started with SBX, even though, it's been a while. And I joked, I said I had a long hair when we started. That's not true, actually. But it's been a while with the technology. But I think, it's kind of always keep innovating. I mean, Niall knows this from so many years at Broad. You keep advancing, keep bringing capabilities, and I just really look forward to seeing what people come up with, how to use SBX.
Right. Now, Niall, again, it's a research tool, but do you have a guess of when this might be something that's used routinely in the hospitals in the future?
Yeah, I think it depends who you ask. I know some of my colleagues at Boston Children's would like that to be ASAP. Um, you
Right.
know, as Mark alluded to, I think Roche has said this technology will become commercially available next year. And so when it becomes commercially available, it will launch as what we call an RUO, or a research use only instrument. And that means that labs will be able to purchase it and use it for research purposes, and that's what Roche will sell it for. However, in the US clinical laboratory setting, we are, as a clinical laboratory, actually able to then purchase a instrument, even in a research use only instrument. And under the CLIA regulations, we as a laboratory can develop what's called a laboratory developed test, which means we validate the use of a set of instruments, reagents, and assays for clinical use. And in our laboratory, then we can offer that as a clinical test under the CLIA regulations. And so that's something that certainly we will be working towards once the instrument is commercially available.
That's great. Now, Mark, you seem to me to be able to see into the future a bit based on what you've invented and discovered. Can you tell us what else you're working on with Roche, with SBX, and other similar or different technologies?
Yeah, I mean, I think there's never a never-ending limit of things you can do. And I think the thing I like about with SBX is it was such a impossible challenge. I think when we started on, all the things that we had to prove what was possible. And I think that's one of the messages I like to get out there is I remember going around talking with people about, hey, this is this idea we have. We're going to take DNA and we're going to change it into something else. And I think just, that we were able to do that in that example of how to innovate. And it was really not easy. It was, so many ways that we could have failed. So much innovation had to happen. I think that's something I think about a lot. And I think about having gone through that, what other innovations we can do. So it's always there, I mean, for me first, I want to get this to market, get this out in people's hands, keep advancing the technology. But yeah, there's a lot of ideas and capabilities that come, in terms of that kind of vision, I think for the future for, empowering researchers to accelerate their discovery, really unlocking complexities of biology, and then really democratizing sequencing. So these are all things that I think a lot about, both in terms of sequencing, but then other aspects of innovation that can be brought to bear, knowing that you can actually do something like SBX.
Yeah, it's incredible. So I want to congratulate all of you for this incredible accomplishment. It's going to make a huge difference for patients. There's no question about that. And I'm looking forward to it being available broadly around the country and the world. I want to thank you all for taking the time to educate us about this important subject so that the average person can understand the significance of this. So thank you very much for your time and knowledge and expertise. We all appreciate it.
Thank you, Rob. Yeah.
Thanks.
Thank you for having us.